Hi friends, today we'll have a discussion on the numerical problems that might arrive in molecular basis of inheritance. The first problem that they have given here is A equals 125 nucleotides, G equals 200 nucleotides. Find the total number of nucleotides and base pair. So the total number of nucleotides will be, say if adenine is 125 nucleotides, it always base pairs with thymine as per Erwin Chargaff's base equivalence rule. That is the reason it will be 125 plus 125, 250. Guanine always base pairs with cytosine. So it will be 200 plus 200, 400. 400 plus 250, it is going to be 650. So the answer for this would be 650. That is what you have to remember about. The second question is find the constant. So 650 is the answer that we have here. So find the DNA constant. The DNA constant in given species is represented by, see, Erwin Chargaff's base equivalence rule, it says that the amount of purine is equal to the amount of pyrimidines. That is, adenine always base pairs with thymine and cytosine always base pairs with guanine. But we don't get a constant there. We'll be getting the answer as A plus G divided by T plus C equals 1. That is not constant for a given DNA segment. The constant, if you have to find the constant, always A plus T should be added and C plus G should be added, and the ratio of this A plus T and C plus G, they are going to give us the constant value of DNA in a particular species. The third problem is, if thymine is 11%, find the total number of pyrimidines. So you should understand that pyrimidines includes cytosine, uracil, thymine. I ask my students to remember it as CUT. And purines are AG. So if thymine, they have given thymine. So in a DNA, if thymine is there, then there is no possibility of uracil. So thymine and cytosine if you have to take. So find the total number of pyrimidines in a DNA they have given here. So you can take the ratio and do it. So thymine is going to be 11%. So adenine will also be 11%, so totally 22%. If you take 22%, the remaining will be the 78%. 78% of cytosine and guanine. If you take divided by 2, so you get 39. 39 plus 11 equals 50%. Or a simple rule you can remember, the amount of purine is always equivalent to the amount of pyrimidines. In any fragment of DNA or segment of DNA, 50% will be purines, 50% will be pyrimidines. So on that basis also you can just say the answer as 50%. Okay. So the fourth one, there are 300 hydrogen bonds and the bases are equally distributed to A, G, C, T. So here it is going to be the bases are equally distributed. So find the total number of bond between adenine, thymine and guanine, cytosine. So we have to find the total number of adenine, thymine and guanine, cytosine. So here what you have to remember is the total number of bonds. They have already given it as 300 hydrogen bonds. I am going to divide it by 5. Because if you know the total possibility of hydrogen bonds, adenine and thymine will be double bond 2. Cytosine and guanine, it will be 3. 2 plus 3 is 5. So if we divide this 300 hydrogen bonds by 5, you get the answer to be 60. So find the total number of bonds between AT and GC. So adenine and thymine, if we have to take 60 into 2, it is going to be 120. Guanine and cytosine, they have triple bond. So you have to multiply it as 60 into 3 is 180. So the next problem is A equals 22%. This is frequently asked, very simple problem. If A equals 22%, thymine is also 22%. So 44% totally they are. So the remaining 56% will be cytosine and guanine. So divided by 2. So 56 divided by 2, you are going to get the answer as 28%. So the next question is, if double-stranded DNA has 102 angstrom length and 20 adenylate, and what would be the total number of hydrogen bonds present at DNA? So you should understand, if you have to find the total number of base pairs, so you have to divide 102 divided by 3.4 angstrom. So we are going to get, so it is going to be around, so we get the adenylate 20. So we have to get the ratio of that. So that 102 divided by 3.4, we get 30 base pairs are there totally in 102 angstrom. So you can see it here that we have calculated that. So now adenylates are 20. So they would be having double bond, isn't it? So adenylates with thymine. So it is going to be 20 into 2. So the hydrogen bonds. So it totally 40 hydrogen bonds as far as adenine and thymidine are there. But total adenylates are 20 and total base pairs are 30. 30 minus 20 is 10. So you multiply this 10, which will be logically, this is the bond between cytosine and guanine. So it is triple bond. So 10 into 3, it is going to be 30. So totally it will be 30 plus 40. So the total number of hydrogen bonds is 70. So that is what you have to remember. This is how we can calculate the total number of hydrogen bonds. Now, this is a formula I ask my students to remember, especially the Michelson and Stahl, where we are going to get this hybrid. You know that in the Michelson and Stahl's experiment, first generation it is 20 minutes, and the newly introduced the bacteria with heavy nitrogen is introduced into a culture media having N14. So the first generation is going to be only 2 of the hybrids will be formed. The first generation. In the second generation, the first 2 generation what is formed, they are going to multiply. So when they are going to separate the strands, so there is a possibility of 2 N14 pure strands, light strand, hybrid strand 2 is possible. Similarly, third generation, it would be 6 and 2. So if you notice here, the hybrid will always be 2. And here they are going to increase in number. See, for every 2 hybrids, they produce 2 normal and 2 hybrids. So that is how the 2 retains up, and the additional 2 is added up into this N14. That is how you get this formula. So here there is a problem regarding 4th generation. If you have this sort of formula, it would be very easy for you to calculate this problem. See, while analyzing the DNA of organism, a total number of 5386 nucleotides were found, out of which the different proportions of DNA base pairs were adenine 29%, guanine 17%, C equals 32%, T equals 17%. If you are finding this sort of proportions of DNA, so what you can consider is, it can be concluded as per Edwin Chargaff's base equivalence rule, the amount of adenine should always be equal to thymine, but there is no equality here. From this we can conclude that the given DNA is not a double-stranded DNA, rather it is a single-stranded DNA. Michelson and Stahl experiment is continued for 4th generation. So I had given you this formula, like which you can remember. 4th generation, there is going to be 14 and 2 hybrids, and light nitrogen will be 14, heavy nitrogen would be 0, or the isotope nitrogen would be 0. So now they have asked you to find out this ratio here. 15 nitrogen by 15 nitrogen, this is 0. 15 nitrogen by 14 nitrogen, the hybrid, here it is 2 of them. 14 nitrogen by 14 nitrogen, it is going to be totally so 2 and 14. So if you reduce that ratio divided by 2, you are going to get it as, see, this is originally, if you put that ratio, it will come it as 0 to 14. So if you are dividing it by the common terms, so it becomes finally the answer is 0, 1, 0 is to 1 is to 7. So a double-stranded molecule have 300 hydrogen bonds. It contains AgCt in equal proportion. So this is a problem which I had already done. So and I told you how to calculate it. So 300 should be divided by 5. We get 60. So the adenine and thymine will be double bond. 60 into 2 is going to be 120. And between cytosine and guanine, so this should be 120, and so this is 120 and this is 180 Cg. So totally we are going to get 300 hydrogen bonds, of which 120 of them are hydrogen bonds found between adenine and thymine. Cytosine and guanine is 180. So adenine and thymine, so this is what we have to remember. Now they have given it as match the following. So number of spirals in DNA. So there is going to be 60. So number of spirals in DNA will be 12 possibilities are there. So for that, how did we come to the conclusion that they have 12 spirals is, see, between adenine and thymine, so 120 hydrogen bonds are there. So 60 is base pairs and this is 60 base pairs. Totally 120 base pairs are there for 300 hydrogen bonds. So if it is 120 base pairs, so when you are converting it into spiral, it becomes as 12 spirals because every spiral has 10 base pairs. So number of nucleotides. So if you take the number of nucleotides, so there is going to be, so it will be, number of nucleotides will be 240. Now you might be asking, how is it? See, there is 60 base pairs. Each base pair has 2 nucleotides. So 60 into 2, 120. Similarly, cytosine and guanine, there is 60 base pair. 60 plus 60. So 112, 60 into 2. So 120. 120 plus 120, 240 nucleotides are the total number of nucleotides present in 12 rings or spirals in DNA is going to be 240 nucleotides. Okay. The next one is length of DNA. So there is 12. So length of DNA, you can multiply it by 12 into 3.4. You are going to get it as 408 angstrom. Number of guanines present in DNA. So already I told you adenine and thymine 60 base pairs, 60 base pairs are there. So obviously number of guanine present will be 60. So this is how you have to find the answer for them all. So the next one, here the answer key, there is a problem here. The answer key is wrong. It should have 120 base pairs as an answer. See, double-stranded DNA shows 6 helices. So one helix is 10 base pairs. And they have 60 adenines. That means 10 into 6 is 60 base pairs. And 60 base pairs has 60 adenines. It implies that they are going to be 60 adenines will be base paired with 60 thymines. So there is no cytosine and guanine here. Total number of hydrogen bonds will be 60 into 2. So that would be around 120 base pairs. So found in that of DNA fragment is, so it is going to be 120 base pairs. So the answer key is wrong. The answer should be 120 base pairs. Simple part of it. If a segment of double-stranded DNA contains 40 base pairs, if a segment of double-stranded DNA contains 40 base pairs, find the number of phosphodiester linkages in the segment of DNA. Usually, the nucleotides have 2 phosphodiester bonds, provided they are linked. Now here it is a 40 base pairs. Within 40 base pairs, so there are nucleotides at both the strands. So they would be having 80 nucleotides. 80 nucleotides implies, so there would be around 80. So 80 nucleotides are there. So obviously it is going to be 80 phosphodiester bonds. But within 40 base pairs, it comes as 39. So 39 into 2, 78 base pairs. So every base pair has 2 phosphodiester bonds. On that basis, 39 into 2, 78. So the answer is 78 of the phosphodiester linkages will be there. It is similar to this. If I'll give you a simple example for one of them. A, sugar will be there. It is phosphate, sugar. So here it would be, just assume C, triple bond G. Similarly, sugar, phosphate, and A double bond T, sugar. So you can find in between the 2 base pairs, within the 2 base pairs, so there is going to be 2 phosphodiester bonds. One is phosphodiester bonds. Okay, so it is going to be in a pair. So the phosphodiester bonds would be there. So you can just look into this part of it. So if you look into 78 of them, it is going to be of 39. Within this, there is 39. So it is going to be 78 base pairs. Always you have to minus it by -2. So the number of CN rings present in a DNA molecule within 2 helices in beta DNA is the number of CN rings. So beta DNA, CN rings implies nitrogen bases. So obviously, if they are having 2 helices, beta DNA, every helix will have 10 base pairs. So as a result of it, they have 20 base pairs. 20 base pairs has 40 nucleotides, 20 into 2. So this is the answer. So calculate the number of hydrogen bonds, that is 20% of adenine is present in 6 turns of DNA. So if you take 6 turns, 1 turn equals 12 base pairs. Helix, if it is 1 turn can be 10 or 12. So here we have to consider, based on the options, we have to consider it as 12 base pairs. Let us assume that it's a ZDNA. So then 6 into 12 is 72. So totally 72 base pairs are there in 6 turns, of which 20% of 72 is going to be 14.4. And if we are going to take it to approximation and put it as 15 instead of 14.4, so we get totally 30 hydrogen bonds, 15 into 2, that is between adenine and thymine. So there is always double bond. So it will be 15 into 2, 30. So we have approximated it. Similarly, when you minus 14.4 from 72, you get 57.6. So if we approximation, if we take, so it would be 58 into 3. So which comes to 174. So 174 plus 30, it gives 204. So the answer is 204 hydrogen bonds. This is how we obtain it. 204 hydrogen bonds. So the next one is arrange in ascending order. Probably the last question of this numericals is arrange in ascending order. So if you take A, number of hydrogen bonds between a pair of It's not between cytosine and guanine. A pair of cytosine and guanine, number of hydrogen bonds between a pair of cytosine and guanine, it is going to be one pair is cytosine and guanine, it is going to be 3 hydrogen bonds, isn't it? Number of base pair in one turn of DNA, 10 base pairs. So type of pyramids in DNA, 2 types are there. One is cytosine and another is thymine. The number of carbon in a ribose sugar, 5. So based on this, on ascending order always is, you should remember the steps from lower step to higher. You are climbing up. So from lowest to highest you have to go. So 2 is nothing but C, 2, 3, then you have C, 2, 3, that is A, D and B. So this is how the arrangement will be 2, 3, 5, 10 based on their numbers of that on that ascending order if we write. So it would be CADB. So this sort of problems can also be asked. Okay, with that we wind up with this numerical problems. If you have benefited from this numerical problems, like and subscribe for the channel and promote my channel by sharing it with your friends. So good luck to your NEET examinations. Thank you.